Alright, so we're going to just start right in with Mutations I. Mutations are simply any acute change in the sequences of nucleotides within the genome. These changes can involve a single nucleotide of the coding strand or many genes. In some circumstances, the rearrangement of vast regions of the genome may occur. The full spectrum of mutations can be categorized into three different areas: genome mutations, chromosome mutations, and gene mutations. Genome mutations occur when there are abnormalities in the normal numbers of chromosomes. Chromosome mutations are the result of rearrangements or alterations in significant portions of individual chromosomes that impact many genes. Gene mutations are changes in the nucleotides within individual genes. This mind map is a screenshot from a Prezi presentation that organizes the different types of mutations. You may find it useful as you begin to explain the genetic mechanisms underlying the genetic disease you have chosen for your topic paper. The link to this Prezi presentation is in your notes. In this lecture, we are going to focus on gene mutations. Again, gene mutations are changes in the nucleotides of individual genes. They can be mediated through chemical and physical agents or occur spontaneously. We will address the specific means by which such mutations are created at a future time. For now, we will be focusing our attention on the relationships between these mutations and the genetic mechanisms that they impact. In this lecture, we will further narrow our focus to a subcategory of gene mutations known as single nucleotide substitutions, or point mutations. With point mutations, a single nucleotide within the coding strand is replaced with another nucleotide. Such a mutation will have a physiological and phenotypical implication if it happens to impact the triplets of bases of the coding strand that correspond with the codons of the resulting mRNA transcript. Recall that the codons of the mRNA code for specific amino acids of the protein that the gene codes for. Also recall that certain codons of the mRNA code for the cessation of translation. These are stop codons. Both of these types of codons can be impacted by changes in their corresponding triplets of bases in the coding strand of the DNA. If these changes result in a change in the meaning of such codons, then the protein product of the gene can be significantly altered. The first point mutation that we will review in this presentation is known as the Missense mutation. With a missense mutation, a point mutation in the coding strand of the DNA for a particular gene impacts a triplet of bases that corresponds with a codon in the messenger RNA transcript for that gene. More specifically, this point mutation happens to change the identity of the amino acid that this particular codon codes for. And here we have our mutation here, symbolized by the lightning bolt. And now we are going to change this single nucleotide, change it from C to G. All right. And this is going to result in the change in the codon within the reading frame. And so the amino acid will be changed from proline to valine. All right. So instead, there is now a missense in the codon as it codes for another amino acid. The result, of course, is that the sequence of amino acids within the protein is altered by the changed identity of one amino acid. If this amino acid happens to be in a location within the protein that is critical for the normal functioning of that protein, such as the active site of an enzyme, the effects on the function of that protein may be devastating. The second mutation we will review is known as the chain termination mutation. Chain termination mutations occur when a point mutation in the coding strand of the DNA for a particular gene impacts a triplet of bases that corresponds with a codon in the messenger RNA transcript so that the codon is either changed from an amino acid to a stop codon or a stop codon is changed so that it no longer acts as a stop codon. In this lecture, we will focus on the former scenario. This particular subtype of chain termination mutation is referred to as a nonsense mutation. This mutation type is called a nonsense mutation because the codon of the mRNA transcript ends up being changed from its original sense. And its original sense is that it codes for a certain amino acid. All right, but this is no longer the case when you have a nonsense mutation. It no longer codes, this codon rather, no longer codes for an amino acid, okay? So therefore, the new codon, which no longer codes for amino acids, is nonsense with respect to amino acids. So that's the context where this term is coming from. Because the codon of the mRNA transcript is changed from one that codes for an amino acid to one that codes for the cessation of translation, the amino acid chain, or the polypeptide, if you will be terminated earlier in its own translation. The result will be an abbreviated or shortened polypeptide. All right. So let's take a closer look at what I've been describing actually looks like. So right here you have the tail end of the coding strand of the beta globin gene. And right over here, where it says Intron, let me get the pen tool on. Right here in this area that's bracketed in with the description Intron, okay, is that part of the coding strand which correlates with what will become the intron in the messenger RNA transcript. And here, highlighted in this aqua color, is that area of the coding strand that will become the exon number 3 within the messenger RNA transcript. Okay, and as you know, in the exons, we actually have the coding sequence, okay, those triplets of nucleotides that correspond with the codons in the messenger RNA transcript, okay? So, and then the way that this diagram is drawn is that all of the nucleotides within the exon, okay, or within the part of the DNA that correspond with what will become the exon in the transcript, in the messenger RNA transcript, are capitalized. All right, and you can see that the amino acids, okay, that correspond with these, that correspond with what will become a codon in the messenger RNA transcript is highlighted above these triplets of nucleotides. Okay, so right here, what I've just highlighted, CTC, is the part of the gene, all right, that corresponds with the codon, all right, which will actually read CUC because we don't have thymine in RNA, right? We have uracil instead. We'll read as CUC in the messenger RNA transcript, okay? And this corresponds with the amino acid leucine, okay? And same thing goes for this triplet and this triplet, okay? And you can see all of their corresponding amino acids above. All right. And by the way, right here, we can see that this is going to be our 5' donor site. This AG represents the 5' donor site, okay, where we're going to splice out, excise out um, uh, the intron, okay, and then fuse exon number 3 to the 3' acceptor site on exon number 2. We'll get more into that later on, okay. Also, within the exon, you can see TAA, okay, which this triplet of nucleotides corresponds with the stop codon in the messenger RNA transcript, okay, and you can see above it, it does not have the abbreviation for amino acid, instead it has the abbreviation for termination. Again, TAA corresponds with the stop codon in the messenger RNA transcript. All right, and although I have it labeled directly here, stop codon, okay, this reference is actually in relationship to the messenger RNA transcript that will result from the information on this exon. Now, again, I have highlighted for you here that the TER represents the fact that this codon, the stop codon, does not code for an amino acid. It codes for the termination of translation. All right. Now, let's focus here on this triplet of nucleotides. Okay. This triplet of nucleotides corresponds with a codon on the messenger RNA transcript that will code for tyrosine. Okay? And actually, this is erroneous. The codon is actually going to be UAU, right? Because if you recall that the codons are actually in the messenger RNA, and RNA does not have thymine. RNA only has uracil instead of thymine. So in reality, the codon is going to read U, A, U, and then this will correspond with tyrosine. All right, but here, what we're actually visualizing is the actual coding sequence within the gene itself. Okay, so this particular triplet of nucleotides corresponds with the codon in the messenger RNA transcript that will code for tyrosine. Now, we mutate the gene. Okay, so this triplet of bases no longer corresponds with the codon in the messenger RNA transcript that codes for tyrosine. Okay, so we end up getting, instead, by changing this tyrosine to a guanine, we end up getting a stop codon in this same location in the messenger RNA transcript. Okay. As a result, translation is going to be terminated earlier. So what's going to happen is we are going to get an abbreviated or truncated polypeptide. All right. Often these truncated polypeptides are innately unstable and dysfunctional. As a result, they are often degraded and make no contribution towards their normal roles. For this reason, nonsense mutations can be especially troublesome. Let's try that again. The result will be an abbreviated polypeptide or protein. Often these truncated polypeptides or proteins are innately unstable and dysfunctional. As a result, they are often degraded and make no contribution towards their normal roles. For this reason, nonsense mutations can be especially troublesome. The third point mutation subcategory that we will review is RNA processing mutations. RNA processing mutations are point mutations in the coding strand of the DNA of a particular gene that corresponds with a component of the mRNA transcript that is either already a splice site or one that can be converted to a splice site. In the first scenario, the point mutation in the coding strand of the DNA results in a splice site in that corresponding mRNA transcript. In the second scenario, the point mutation in the coding strand of the DNA results in the creation of a new splice site in the corresponding mRNA transcript. We will first turn our attention to the latter scenario, where the mRNA transcript is altered by a point mutation so that a new splice site is created. In this situation, as is the case with all the rest in this presentation, the point mutation occurs in the coding strand of the DNA. However, this alteration in the coding strand of the DNA happens to result in the creation of a new splice site. So now we're taking a little bit of a bigger view of the beta globin gene, and we can see all 3 exons. We can't see the untranslated regions, right? So there's no regulatory regions visible. We're just seeing actually part of exon 1, part of exon 3, and you can see exon 2 has been clipped right in the middle so that it's abbreviated. All right. And as always, okay, when you see the uppercase nucleotides, that means that those are the nucleotides that are components of the coding strand. Okay, so they're going to be involved with, coding for amino acids, or the start of translation, or the end of translation. All right. So at the top, you can see normal splicing, where exon 1 and exon 2 and exon 3 are cut out. Okay, and then they're fused together. Okay, and then you have your final mature mRNA transcript right below the blue arrows. All right, and then you can see that the splice sites are highlighted in red. Okay, on the sides of the exons that we can see. All right, so let's just do some recall first. As you recall, splice sites come in 2 different types. The exon intron splice sites, or the 5' donor sites, occur at the left-hand side, okay, or the 3' side of exons. All right. So actually, let's just go ahead and we can draw in our 3' side, okay, of these molecules, of these DNA coding strands. Okay, so over here to the left, we have our 3' side, okay, and then over here to the right, we have the 5' side, okay. The splice sites that are to the left of the exons are the exon intron splice sites, or 5' donor sites. Okay. This is the site of which is donated to the 5' side of an adjacent exon when the introns are removed. All right, it's called the 5' donor site. All right, because this is the side of the exon that is then donated to the 5' side of another exon when they are fused together to form the mature mRNA transcripts. Okay. All right, so the intron exon splice sites or 3' acceptor sites occur at the right-hand side or 5' side of exons. All right, the side of which accepts the donation of the 3' side of an adjacent exon after the introns have been removed. All right, so again, we're going to redraw our 3' and 5' sides. Oh, let me first, I guess I got to go down and select the pen tool here. Okay, so again, this is the 3' side, okay, and over here is the 5' side of this coding strand of our DNA molecule. All right, and so the intron exon splice sites, okay, are going to be on the right-hand side, okay, or the 5' side of these exons. Okay, so this is going to be the intron exon splice site or 3' acceptor sites. Okay, and why is it the 3' acceptor sites? Because this site will accept this side of the exon will accept the 3' side of the adjacent exon when they are all fused together into the mature messenger RNA transcript. Okay, that will be translated. All right, so these terms are not ideal. It's very confusing. So it's probably worth a moment for you to just stop the video now and look at your notes and just write down these terms about 10 to 20 times over with their definitions. And there's nothing wrong with memorization. And take the time to mark up your notes just like I'm marking up these slides. All right. So on the animation sequence here, first we see that the point mutations result in the creation of a new exon intron splice site or 5' donor site. Okay. So right here, the lightning bolt represents the mutation. We go from guanine, okay, or I'm sorry, rather we go from adenine to guanine, okay, and ta-da, we made a brand new 5' donor site, okay, or a new exon intron splice site, okay. This will extend the length of the original exon number 2 to its left-hand side or 3' side, okay. As a result, exon number 2 will contain more bases and more codons. When the abnormally long exon number 2 is incorporated into the mature mRNA transcript, these extra bases and codons will also be incorporated. Also, on the animation above, we see that the second point mutation results in the creation, here's the second mutation, symbolized by the lightning bolt. Okay, we see that the second mutation results in the creation of a new intron exon splice site, or a 3' acceptor site. Okay. So we used to have an A, an adenine, now we have a guanine, okay? And there you go, GT. That is a 3' acceptor site, a brand new splice site that we just created. Okay, this will extend the length of the original exon number 2 to its right-hand side. As a result, exon number 2 will again contain more bases and more codons. When the abnormally long exon number 2 is again incorporated into the mature mRNA transcript, these extra bases and codons will also be incorporated. So in both of these cases, okay, whether you're creating a new 5' donor site, splice site, or a new 3' acceptor site, splice site, some of the normal sized exon number 2 will be excised from both of its normal splice sites because both of its normal splice sites still remain intact. Okay? So again, let me show you this. This illustration right here at the top shows you, reiterates to you rather, that this normal messenger RNA will be translated into a normal protein. All right, and then here, when we create a new exon intron splice site, okay, or 5' donor site, okay, we can see now that we get a little bit of the extended exon number 2 mRNA transcript, okay, but at the same time, the old splice sites are still there, so we end up getting a little bit of the normal mRNA transcript, okay, that we see up at the top there, which will result in a normal protein. All right. Okay. And then we see right here the translation of the extended exon number 2 mRNA transcript results in an abnormal protein, okay, after translation. And then again, that the normal mRNA transcript, okay, that results from splice sites that are normal, okay, continuing to be effectively utilized, okay, will result in an mRNA transcript that produces a normal protein. All right? However, because we do have the addition of these new splice sites, okay, the new splice sites, whether they be in the 5' donor site or the 3' acceptor site, will compete with the normal splice sites during the process of mRNA splicing. All right, so that same thing is going to be going down here, okay, where we have both the extended exon number 2 mRNA transcript, okay, competing with the normal mRNA transcript. All right? And so because you see competition between the normal and abnormal splice sites, you get a little bit of the abnormal protein and you get a little bit of the normal protein translated from these alternative transcripts. All right? So the big picture is as a result of all this competition from the abnormal mRNA transcripts that result from the abnormal additional splice sites and the abnormally long exon number 2s, there will be a decreased amount of normal protein translated. Okay? So again, keep in mind that the enlarged abnormal mature mRNA transcript with the enlarged exon number 2 will not result in the translation of viable proteins. So as a whole, you end up getting a diminished amount of translated viable protein. Now we will turn our attention to the previously mentioned scenario where a point mutation in the coding strand of the DNA results in the eradication of a splice site in the corresponding mRNA transcript. All right. In this circumstance, a splice site that is usually present in the mRNA transcript of the gene is no longer present as a result of a mutation. As a result, one of the boundaries of an exon within the initial mRNA transcript cannot be defined. This prevents the affected exon from being incorporated into the mature mRNA transcript. On the animated sequences, first we see that the point mutation results in the elimination of the exon intron splice site. Okay. Oh, and by the way, up at the top we have normal splicing shown, so we'll just add that in like we did before. You're all familiar with the way splicing is supposed to work on this beta globin gene. All right, but now we're going to turn our attentions to a point mutation in the exon intron splice site, or 5' donor site. Okay, so there it is. There's the lightning bolt, and it's changed from adenine to cytosine. Okay, and so instead of reading AG and being a normal 5' donor site splice site, it now reads CG. Okay, because of this change, exon number 2 cannot be fully excised out of the initial mRNA transcript. Okay, this is going to result in a truncated mature mRNA transcript that only possesses a fusion of exons number 1 and number 3. So also on the animated sequence here, we see that the second point mutation results in the elimination of the intron exon splice site, or the 3' acceptor site. So here we have the 3' acceptor site here, GT, and we see the lightning bolt takes out thymine and turns it into adenine. So before we had GT, which was an acceptable 3' acceptor site splice site, and now it is no longer. All right. Because of this, it is again apparent that the exon number 2 cannot be fully excised out of the initial mRNA transcript. Okay, this then results in a truncated mature mRNA transcript that only possesses a fusion of exons number 1 and number 3. In both cases, the abnormal and truncated mature mRNA transcripts that lack exon number 2 will be missing substantial amounts of codons. As a result, they will result in the translation of grossly abnormal proteins that will be missing substantial amounts of amino acids.